Hi, everyone. Welcome to Process Biotech and Health Extension Group. I'm really delighted to have all of you here. And I'm so delighted to have Alex here today, who is now a fellow alum, I guess, and has been joining Foresight previous workshops and Vision Weekend and so forth in particular, to discuss the role of the ECM in aging. Thanks for wearing your Foresight t-shirt. I love it. I'm wearing a Foresight pin too. So thank you so much for joining. I know that you've been doing absolutely wonderful work in this area. And we're so delighted to have you on today to discuss this topic in depth, given how much recent attention it has received and given how long-standing your work in this area has been. So I guess you should kick us off with an intro, and then we'll be moving to the Q&A later. Feel free, everyone, to drop your questions in the chat. I'm sure there'll be plenty, and I'll be moderating and perhaps asking you to hand intro questions after you presented. So thanks a lot. Thanks a ton for joining, Alex. The stage is yours. Take it away and hi, everyone. It's good to see everyone.
Thank you, Elastin. Thank you for having me here. I'm really pleased and it's an honor to present here for such an audience. And yeah, so my name is Alexander Fedintsev. I'm a biogerontologist, and I'm the author of the paper that proposed aging of long-lived molecules, especially in the ECM extracellular matrix, as a 10th hallmark of aging, which was long time overlooked. And here I explain why it is so important, but more than that. So I'll try to convince you that it might be not just a 10th hallmark of aging, but it might be a key driver and the most important reason of aging, like cause of aging. And I always like to start my presentations from the quote of one of the most intelligent human beings ever lived on the planet Earth. This is Richard Feynman, very famous physicist. And he said once that there is nothing in biology yet found that indicates the inevitability of death. And this is a wonderful quote. So if the most intelligent person has small doubts, like very little doubts about the possibility of indefinitely extended human lifespan, why should we have these doubts? And yeah, basically, what limits our possible indefinite lifespan? Of course, aging, right? Aging is the most difficult obstacle of reaching biological immortality, because we all know that the risks of death increase exponentially. Roughly 8 years, the risk of death from all causes doubles, which basically limits our lifespan. And it still is widespread hypothesis that this is due to death of cells, that they start to functioning not properly and they die, they cannot live more than 120 years or so, or they're programmed to die at certain age. But then, I was thinking about it, what causes aging, for a very, very long time, so maybe more than 10 years. And 5+ years ago, I realized that cells have all the mechanisms in place to live indefinitely long. Why? Because they have autophagy, mitophagy, DNA damage reparation systems. They have proteasome, whatever mechanisms to keep all the machinery in place in a fully functional state. But what causes aging then, right? That's a big question. And this, my hypothesis, I started to basically study a lot of literature and papers about the capabilities of cells, how long can they live. And to my surprise, or not, it turned out to be that cells can outlive the maximum longevity of various organisms, several fold at least. For instance, this very, very old paper claims that basically hematopoietic stem cells that give...
Do you want to screen share, or are you not screen sharing yet? Just want to make sure.
I'm not screen sharing. Okay.
No, not at the moment. And
okay.
So far, everything was understandable. But now that you're discussing your paper, I think you were...
Yes. Yeah. Oh
Great.
my God.
This is
I'm
good.
sorry. Can you
No,
see
we
the
all got the
screen?
quote. Yeah, this is great.
Awesome. And then, well, basically, this paper claims that mouse hematopoietic cells that give rise to all the blood cells can live up to 3,000 days, which is 2.5, more than 2.5 fold like of the maximum longevity of mice. This is like incredible. And there were no like limits of longevity. So perhaps it is possible that these cells can live even more. But one can say, okay, this is like pretty old work, so maybe it's like maybe there was some biases or errors, whatever. There is another paper, very recent paper, where researchers did the same thing with serial transplantation of T cells, T lymphocytes to mice. And they found exactly the same, basically, result. So they found out that T cells can live like virtually indefinitely long. So being serially transplanted to mice. And of course, this is not limited only to blood cells. There is another study, very interesting, where like mouse brain cells, scientists have genetically engineered mice, which have green fluorescent protein expressed in their cells, so they can visualize them. And they implanted these brain cells into brains of rat embryos, and these cells successfully integrated into the brains of rats, and they live like for up to 36 months, while the maximum lifespan of these donor mice was only 26 months. So it's almost 40% more than the maximum longevity of the organism. So we see clearly that cells can live much longer, different types of cells. There are dividing cells like our blood cells, lymphocytes or stem cells, and also like post-mitotic cells like brain cells. And we see clearly that cells clearly have capabilities to live longer than the organism as a whole. And that's why, so I hypothesized that it is the aging of something that is not repairable that drives aging, that basically initiates this self-propelling vicious cycle of damage and destruction. And well, basically, I started working on the paper that basically brings all the evidence together. And yeah, I got a like extremely valuable comments from Aubrey de Grey and extremely like important help from my co-author, Professor Moskalov. And together we issued this, like we finished this paper, published it, and you can read it. But like here in this paper, Aubrey proposed to call it like 10th hallmark of aging, missing hallmark of aging. But now I think that it is even bigger than that. So it's not just a hallmark of aging, it is probably one of the true causes of aging. And I call it strong ECM hypothesis. Basically, and now let's discuss mechanisms that basically kill us. So what happens with ECM and how it well causes different pathologies and eventually death. So, and one of the most important like long-lived protein in our body is elastin. Elastin is a structural component of blood vessels, skin, lung tissue. And the interesting thing about elastin is that it is deposited at infancy, basically. And then stays virtually untouched. So there is no turnover of elastin. There is no synthesis, like virtually no synthesis. There is some, but it's like minuscule. And here we can see what happens to the elastic fiber. This is a very famous picture, by the way. We can see what happens to the elastic fibers of like extremely old people. On the left, we can see young elastin, undamaged, basically very nice fibers. And here we see like a very damaged elastin, which loses its main property to basically to give elasticity to tissues. All right. So yeah, there are lots of mechanisms of elastin damage because it's not renewed, and like various damages like mechanical stress primarily, then oxidative damage and matrix metalloproteinases, all them destroy this elastin mesh that basically provides elastic recoil. And basically what's the result of that, right? Well, of course, result we see on everyone's face, right? With aging, we lose elasticity, we start to accumulate wrinkles. It's not very super like appealing to have wrinkled skin, but there is more to that. So elastin helps us to breathe because lungs contain a lot of elastic fibers, and we need to exhale. We don't need to basically work. Like our muscles do not need to contract because air is exhaled just by elastic component of lungs. So when elastin is gone, so it's much difficult to breathe. But the most important function of elastin is, of course, in cardiovascular system. So our main artery called aorta is an elastic type artery. It contains a lot of elastin, and it provides dampening of the blood flow. I will explain in a moment like why is it important. Yeah, so basically there are some pictures and then diagrams which are not very illustrative, but I have something that basically that can explain what I mean very, very intuitively. So the mechanism by which aorta protects our organs is based on an effect called Windkessel effect. It's, I guess, from German. And here is the video how it works. Let's spend a little bit time. Do you see the video?
Yes.
So we can see that here we have like pretty aggressive blood flow, well, not blood flow, but water flow, which hits the wall of a box, right? Hits pretty hard. But let's see what happens if we add some elasticity. So blood flow now is more steady. It's less. It doesn't hit that hard the walls of the box, and this is exactly what happens with our organs and tissues. So the aorta provides this dampening of blood flow, and when we lose elasticity, this blood flow becomes like a real problem because it damages these gentle capillaries in various organs and tissues. And it is very well known that these rigid arteries, the arterial stiffness, is linked to kidney damage, brain damage, like various organ damage. And there were studies where artificial stiffening of like arteries that feed brain leads to dementia in model organisms. So it is extremely important to keep our arteries elastic. And there is a paper basically that claims, and I cite this paper often, that elastin degradation puts the limit on human maximum longevity. And from the picture, we can see that basically at a given rate of decline of elastin in arteries, there will be virtually no elastin by the age of 120. And it means that basically human organism cannot exist given this rigidity of arterial wall. So basically this is one crucial bottleneck that limits our maximum longevity. But this is not the only thing that well, in the ECM that can be damaged and can negatively affect our health and longevity. Let's talk about collagen, another long-lived protein that comprises ECM. And yeah, basically, we know that ECM is tightly connected to cellular senescence and basically stem cell characteristics and whatnot. There are studies that show that like old stem cells placed on young ECM basically gets rejuvenated and expand as young cells and so on, and vice versa. So young cells placed on old ECM will behave like old stem cells. Yeah, there is certain connection between cell functionality and the ECM quality. And yeah, but what are the features of old ECM? One of the most studied and most important mechanism of ECM aging is accumulation of so-called crosslinks. These crosslinks are a result of a spontaneous reaction of glucose and other reducing sugars with long-lived strands of collagen. And we can see there are two types of different modifications that spontaneously arise during like our regular life. So there is no need for enzymes, there is no need for some machinery. They just react and stay for very long time. So there are two things: crosslinks and adducts. We will talk a little bit later about adducts, but let's focus on crosslinks. So it seems that nothing important is happening. So, well, crosslink and crosslink. But in fact, these crosslinks, they change the physical properties of collagen. So first of all, it becomes much stiffer. And in my review, you can read about like a lot of bad things that stiffened collagen can cause. But now we will talk about the second thing that is caused by this cross-linking. So cross-linked collagen becomes less susceptible to the enzymatic degradation. So in normal tissues, collagen is subject to remodeling. So there is a constant turnover of collagen. There are enzymes that cleave collagen, dissolve it, and there is new collagen deposited all the time. So this process is like ongoing. Of course, it's not very fast, but it is ongoing. So the half-life of collagen is years in comparison to elastin, which is like tens of years or even hundreds of years. Then basically what cross-linking does, it basically cross-links collagen and prevents collagenase enzyme that cleaves collagen to sit on the like molecule, find the site, cleavage site, and cut the strand. So these crosslinks prevent that. And as a result, we can see like here, very, very illustrative example. So here we see on the picture on the left, we see 3 rat tails. One rat tail was like control, and the rat tail in the middle was basically incubated in the solution of ribose. This is a sugar, and for 3 days. And the tail on the right was incubated in ribose solution for 7 days. And what we can see, and then these rat tails were placed in the collagenase solution, so enzyme that breaks collagen. And what we see, like after 5 hours, the control rat tail was basically half dissolved, and after 25 hours, almost entirely. But the one that was incubated in ribose for 7 days is intact. So it basically very, very resistant to this collagenase. And of course, this is in vitro experiment, but the same happens to us human beings. So this is also from quite an old paper. And we see that with age, the insolubility of the collagen grows non-linearly. Let's put it this way. But we see the very tight correlation between time to dissolve collagen by collagenase and age. So very tight correlation, probably maybe as tight as epigenetic clock. But it's like we need more data to say. But okay, what's the problem? Okay, collagen becomes less soluble. What's the problem? What's the bad thing about it? And it turns out that there is a very, very important problem. So here is the study where researchers try to mimic what happens with collagen during cross-linking. So collagen becomes less soluble, and they imitated this by creating mice which collagenases were altered by mutation. So this collagenase cannot efficiently cleave normal collagenal mice by effectively mimicking this collagen insolubility situation. And what happened? These mice became progeric. So they basically developed accelerated aging, like dramatically accelerated aging. And what is most importantly, these blood vessels of these mice contain a lot of senescent cells, which are stained in this blue color. I'm not sure what color it is, but you can clearly see it. So in wild type mice, there are like virtually no senescent cells, but in this mutated mice at this very young age, there are tons of these senescent cells. So there is a clear link between cellular senescence and ECM properties. And like what is the widespread view on cellular senescence? Well, most researchers think that cellular senescence is a mechanism of cancer protection from cancer. Basically, that cells stop to divide due to shortened telomeres and so on and so forth. But this explanation, I think it's not very good. Why? Because we know that mice when senescent cell clearance in mice also reduces frequency of tumors. It prolongs life and postpones the onset of cancer. Senescent cells appear to be pro-cancerous. And this is like a huge problem in this hypothesis. But more to that, there is a growing body of evidence showing that cellular senescence might be an anti-fibrotic program. And this makes total sense because, what the senescent cell the main feature is, senescent cells are known because of so-called SASP, senescence-associated secretory phenotype, which includes such molecules as pro-inflammatory cytokines and, very important, matrix metalloproteinases, which are supposed to dissolve like excessive deposits of collagen, which makes total sense in theory, like in light of this hypothesis. But why do we accumulate like senescent cells with age, and how it is connected to the ECM alterations during aging? And this is my primary hypothesis that I've stated in the article, that this glycated ECM, by some physical properties, stiffness and solubility, mimics the fibrotic ECM, and thus causing this anti-fibrotic response in the form of cellular senescence. And I call it systemic pseudofibrosis because there is no excessive collagen deposits, but cells think there is fibrosis. And so then this is basically, but this is not limited to cellular senescence, this collagen insolubility problem. So let's Let's think about brain. So what happens in brain? There is a very, very important thing called neuroplasticity. It's the ability of neurons to form new synapses, new contacts with another neurons. And it happens to be that to form these new synapses, neurons need to clear debris of brain ECM, basically using very similar enzymes, but the molecules comprising brain ECM are slightly different than collagen. But anyway, there might be cross-link accumulation there as well, and also, like, consequently, there might be decrease of solubility of the brain ECM molecules, and it may hamper the ability of neurons to form new synapses, because it's harder to dissolve this debris, to clear this debris of matrix in brain. And well, it might very well explain like decline in neuroplasticity during aging. All right, so let's talk about the next like crucial thing that is also might be connected to the ECM. It is called clonal hematopoiesis. It's a pretty well-known thing among hematologists, so people who study blood, but it was also a little bit less popular in biogerontologists. Like very few people talk about it. Yeah, but it turns out that it can put another limit on our maximum longevity. And yeah, so I'll explain a little bit about this clonal hematopoiesis, what's that, and why it is so important. So what is age-related clonal? Yeah, so I basically knew this like long time ago when I read about like super centenarian woman who lived till 115 years or so. And interestingly, her blood cells were descendants of a handful of clones. So normally your blood cells are very diverse. They basically like multiple different stem cells produce them. But it turns out to be that with aging, this diversity decreases. So we can see, for instance, that a frequency of specific mutation in blood in this woman is very high. So it means that there are a lot of cells which basically born from one or several clones, right? And this very similar situation in lungs, but it is not so dramatic. Okay, that's the thing. But interestingly, this happens to all of us, not only centenarians. So this prevalence of clonal hematopoiesis increases with age at a faster than linear rate. So I'm not sure whether it is exponential or not, but it's definitely very fast. And if you can see here, so if you can look at the picture, we can see that almost half of people aged 85 to 110 years have this clonal hematopoiesis. And probably like people older than 110, like probably 90% or 100% have this thing. And why is it so important to basically pay attention to this problem? Because clonal hematopoiesis is not that benign at all. So why is it happening? Because these clones apparently that basically survived and expanded numbers, they are less lymphoid biased. So what does it mean? It means that we have, when we are young, we have slight bias to lymphocytes to produce cells of adaptive immunity that can learn new infections, new antigens, memorize them, and then efficiently counteract different infections and so on. And it turns out to be that with aging, there is a myeloid skew. So we have less adaptive immunity and more innate immunity. And this is not very good by itself, because adaptive immunity is crucial for immune response, for instance, against influenza or COVID-19 or other infectious diseases. And innate immunity is not an innocent is not an innocent thing. Like these myeloid cells, they are very, very pro-inflammatory, and it increases inflammation further, like contributing to this inflammation and have a increase in the likelihood of atherosclerosis, or contributing to the pathogenesis of atherosclerosis. Clonal hematopoiesis is associated with poor survival and risks like 20-fold increase in cancer and blood cancer risk. So it's certainly a life limiter. And it is very important to understand why is it happening. Well, first ingredient to clonal hematopoiesis are, of course, mutations. Spontaneous mutations, which is a normal, absolutely normal thing that happens with dividing cells, but these mutations are basically random. And why some cells gain competitive advantage? It's because of the second ingredient, the environment that changes with aging and favors certain clones harboring certain mutations. And there are indeed certain genes where these mutations are more likely to be detected. And these mutations basically cause this like more rapid proliferation, but also this myeloid skew and worse functioning of blood cells. And what's in the environment that causes these specific mutations? And it turns out to be that the key factor, the common factor, is inflammation. So certain inflammatory cytokines are linked to this clonal hematopoiesis. And well, basically, what about like how we connect ECM and inflammation? Well, first of all, we remember that this cross-linked ECM may cause cellular senescence, and senescent cells, they basically secrete these pro-inflammatory cytokines, and this could be one link. There is a more direct link, more apparent link, and it is, so this is the mechanism, how, like, putative mechanism, of course, how this clonal hematopoiesis arise. Yeah, but this is the most important bit. So, like, we talked about crosslinks, but there is another type of advanced glycation end products. They're called abducts. So they're like free hanging advanced glycation end products, which can stimulate so-called RAGE, receptor for advanced glycation end products. And this RAGE, this receptor, is known to be very, very pro-inflammatory. It is basically a component of innate immunity. So this constant stimulation of RAGE by abducts may create this perpetual self-propelling, positive feedback loop of inflammation that in turn drives clonal selection and age-related clonal hematopoiesis. And it might happen not only with hematopoietic stem cells, it might not only happen with blood cells. There are tons of other places like colon or lungs, as we saw previously, maybe microglia and brain, where this clonal hematopoiesis might occur. So this is basically 3 previously unknown mechanisms by which old ECM could potentially kill us. So there are a plethora of other mechanisms. You may read about them in our review, our paper about 10th hallmark of aging. Yeah, so a couple of words about people who want, and sorry, who want to change the situation and who are aiming to reach like longevity escape velocity eventually. This is our team, a group for radical life extension. Our goal is basically reaching radical life extension, which we define as at least two-fold increase in expected lifespan. But ultimate goal, of course, is to reach longevity escape velocity. And we're basically a community of scientists, entrepreneurs, and biohackers who are known for trying for the first time this plasmapheresis in humans. We're doing tons of other experiments, but we're also developing gene therapies against aging, and we also focus on two of three things that I described, the two of three these pillars. We are focusing on elastin remodeling and combating clonal hematopoiesis, because we think that probably cross-linking is a little bit difficult problem for current level of technologies. But nevertheless, we believe that fighting at least two or three problems can give us a dramatic increase in life expectancy. And yeah, so if you want to participate with us, help us in some way, please contact us. We will be happy to collaborate, and let's fight aging together. Thank you.
That was awesome. Thank you so much. Really useful with the last slide as well. We have plenty of questions already in the chat. This is a reminder that you're welcome to put your questions in the chat or just raise your hand, and we'll start with questions from the chat. Alex, you had a lot of questions, so if you're interested in kicking us off, feel free. Otherwise, I have Micah and Carl next.
Sure.
Well, maybe I'll read out one question from Alex, and then maybe you can get back to it. Can mechanotransduction of excess stiffeners be part of the signal that reaches the epigenome and nucleus? And can mechanotransduction inhibitors, for example, YAP, reduce some of the effects of ECM damage?
Yes, for sure. Mechanotransduction is a transfer to nucleus and was shown to affect both histone modifications and methylation. So there is a certain connection with it. We don't know like what is the degree of this influence in epigenome, like age-related epigenome modifications, but there is certainly some contribution of ECM to epigenetic landscape during aging. That's undoubtedly. And yes, these inhibitors like ROCK inhibitors, and by the way, for instance, a very good example of this are statins. Statins are they have off-target effects, so they inhibit this mechanotransductional pathways, so they block ROCK, if I'm not mistaken, and they are known to decrease all-cause mortality by as much as 20%, if I'm not mistaken. So there is like a very good example. We don't know, of course, why they work, but presumably, at least partially, they decrease all-cause mortality by interacting with mechanotransduction pathways. Because just by lowering LDL and their effects cannot be explained, because there are plenty of other medications that lower LDL and do not yield such a big increase in lifespan.
Wonderful. Then I have another question from Alex, and feel free to drop more questions here in the chat or raise your hand. Are small amounts of collagen per unit time in cross-linking reversible in young animals only, and are larger amounts less reversible?
Could you repeat? So small amounts of collagen
are small amounts of collagen per unit time in cross-linking reversible in young animals only, and are larger amounts less reversible?
Yeah, that's a good question. Yeah, it all boils down to the pace of turnover of collagen. So if in theory we can basically degrade collagen faster than we accumulate basically these crosslinks, then it might work. I cannot answer precisely the question, are these small amounts of cross-link reversible or not? But there is certainly some good ideas in this field. So there is certainly some rationale behind that. So yeah, we need more studies. So let's put it this way, and we need more funding in this area to do that.
Wonderful. Next one, we have Jean.
Oh, was I next? I thought, hey, Alex.
Nice to see you.
I thought Micah was ahead of me. Is it?
Micah just wanted to say his kudos to Alex's presentation.
Okay. Yeah, great. Yeah, nice to see you again, Alex. And
Likewise.
again, like super important. I think you did a great job here at showing why more of us should be focused on these issues that are the extracellular parts of our body rather than just the intracellular parts. With the underlying implication, given all the studies that you showed here and others, that really this focus on cellular processes are going to have a limited benefit on extending maximum lifespan. And the other point I think that's important here is that you mentioned a little bit with collagen, but also with elastin, right? There isn't much repair, or in some cases, there is no repair for this kind of damage that occurs extracellularly. So even these sort of imagined gene therapy approaches might be difficult not only to implement, but to come up with new genes capable of doing these things. But the question I had actually was more to do with what I haven't found any studies on this yet with the extracellular environment. It's not just made of proteins, right? It's also made of carbohydrates, especially in the brain, which is on a little bit. And no one's even really, as far as I can tell, started looking at that and the damage that accumulates in these long carbohydrate chains.
Yes.
That in a nice, warm, aqueous environment over the course of decades, you might expect also accumulate damage.
Yeah.
Do you know any good studies that have tried to look? It's very hard to look at the glycome.
Yes.
Because it's much more complex than the proteome.
Yeah.
So I don't know if you know of any, but I have not found any. So if you know of any, I'd love to hear it.
Yeah, that's a very good question and like a very good point. So when I thought like about the insolubility of the ACM in the brain as a cause of this reducing neuroplasticity, I also questioned myself that, okay, like brain is comprised of it's supposed to comprise of proteoglycans, right? And the only thing that I was able to find was a study that basically showed that crosslinks these ages accumulate in cartilage proteoglycans, which is not the same, but at least the class of molecules is the same. I can share the link with you if you want. So
I think I know that one. Yeah.
Yeah.
I was thinking specifically.
Yeah. In the brain, unfortunately there is no, and this is completely like unexplored region. So where we can basically find a lot of new insights into the aging of brain, right? Because what we are now focusing is these amyloid plaques and tangles and whatnot, but the true cause might lie outside of the cells, right? So there is like structure called perineuronal net, which is one of the examples of brain ECM, and it is known to participate in synapse formation, right? So it's pretty important, and we don't know what happens with it during the course of aging. Who knows?
Yeah. Yeah, I think we do know that it stiffens, but we don't know the components.
Yes.
Great. Thanks so much.
Thank you.
Cool. Next up, we have
what's the status? Actually, I have two questions. What's the status of RLE group? Like, it's a for-profit, it looks like. You've got multiple projects. Not all of them are ECM. The ECM project doesn't have a more info link. I'm assuming that's just early in that project. Do you actually have a therapeutic approach yet, a mechanism of action identified?
Yes. So basically,
talk about it, or is it stealth?
Yeah, I can expand a little bit more on that. So RLE group is initially a non-profit organization, right? So basically, we are trying to reproduce various interventions that are very promising in extended lifespan, but also we are developing several like therapeutic approaches on our own, and like some of them will be for-profit organizations, startups. We don't have registered them anywhere yet, but we have something working. So we are working on gene delivery system, and one of the application of this gene delivery system is to basically de novo synthesis of elastin, right? And the very obvious place where we can do that and we can get tangible results and is skin. So we can overexpress elastin and fix the wrinkles and then go to like cardiovascular problems and so on and so forth.
So the parent is a nonprofit and you haven't spun off any of the for-profits yet.
Yes.
Where have you gotten the money?
We have donors. So
and what country is it based in?
So, well, basically we are like our nonprofit is in Spain. Yeah, some research is happening in Russia currently, but we are thinking about moving somewhere.
Okay. So question number 2, if we could solve everything in aging except ECM stuff, if everything else got solved, all the other hallmarks, all the SENS damage areas, what do you think maximum human lifespan would be?
120 years.
But that graph you showed, which kind of pointed to 120, was what currently happens, right? It was an extension of what currently happens in the context of, but the ECM presumably will age slower in the context of persistently young cells. So shouldn't it be longer if we solve all the rest of aging?
The problem is that it doesn't depend. Well, it depends to very little extent.
Because older cells still turn out
ECM
proteins.
So basically, it declines at a constant rate. The rate of decline of the elastin, for instance, the damage to elastin is the same at younger age and at like very old age. So if we look at pulse wave velocity, which is a marker of stiffness of arteries, it grows linearly with age, meaning that there is no acceleration, or the acceleration is so tiny that we cannot detect it. So it means that even if we keep everything young, right, but we have this ECM problems like cross-link accumulation and elastin damage, we won't leave, like our maximum longevity won't increase like much, because you saw this mice genetically engineered with collagenase that were incapable of dissolving collagen. They did not have any other problems. They didn't have any DNA damage or whatever. They basically lived 4 times less than their counterparts. So the only collagen solubility problem basically limits the longevity, and it doesn't depend on cells at all, because it depends only on the concentration of glucose, maybe a little bit on temperature, and on the collagen itself. So it's not controlled by our bodies, and this is the problem. So we can't do anything about it by targeting cells, unfortunately. And the elastin problem with elastin, that it is remodeled only, like not remodeled, but it is created only at infancy, and that's it, and then it stays the same. And this is the problem. So even if you rejuvenate cells, this is nothing to do with elastin. It continues to decay, to basically the destruction of it happens just by mechanical oscillations. It ruptures, fractures bit by bit, slowly but steadily, and it doesn't depend on anything else. So of course we can slow down a little bit by like lowering the elastasis, lowering the oxidative stress in arteries, but this is like a minor component. We probably slow the degradation of elastin by 5% at max, which doesn't increase lifespan by a large amount of years. So it will be somewhere, like my ballpark estimation, it will be somewhere around 120. Given that currently, I don't believe that we have a person that lived past 115 years.
Interesting. Okay, thanks for that input. Well, we only have 3 more minutes left, and I do want to make sure that I ask my standard question, and which is that if people are excited about supporting your guys' work, what are a few next steps that you and your organization are about to take? How does your, I don't know, next year or something look like ideally? And what could people do to kind of like help, collaborate, support, anything like that?
Yeah, we have a few very interesting, promising projects that are not very, very long to execute till like first results, and we would like to, well, start working on them somewhere in some location. So register a company, get some start, and yeah, basically do something practical about it.
And if people were interested in helping you, how would they best, like they would just reach out to you via email? Are you looking for specific collaborators? Are you looking for specific funders? Are you looking for, I don't know, like, are you hiring or, are there specific bits that people could follow up?
Yeah, that's a very good question. So basically, any help is appreciated. Any help, but of course, funding is number one because it's very difficult to do anything like in this field and in biomedical field without funding. But also, like other help is also very, very. It's priceless. Like legal, like consultations. If someone with biological background can help us like with anything, that would be like really, really priceless. Yeah, our mission, as I said, is basically to defeat, not to defeat, but radically extend longevity, and we are doing it on our own, all right, but we need more help from the community to really speed up things, because we're already
Wonderful.
at a pretty good pace, but we can do much better.
Yep, we definitely have to move a little faster. Well, thank you so much, Alex. This was really wonderful.
Thank you.
Thanks so much for being a part of the project community. I remember coming across you on the internet and I couldn't believe what you were working on. And I'm really glad that you listened to my cold email outreach like 2 years ago, and really excited to have you as part of our community. Thanks a lot for presenting this really wonderful work. Thanks everyone for joining. It's been very fun. And I'll see you guys at the next one. Thanks everyone.